So welcome back. This is lecture 7. I can't believe we're getting already to lecture 7 in this course. Today, we're going to talk a little bit about the immune response at viral infection, and I'm going to be using a set of slides that were prepared by a master's student last semester, Sam Heaps, in the class I taught in the spring. He gave 2 lectures on the immune system and put together a very nice set of introductory slides on viruses and the immune response. They may be a little bit more detailed than we need today. So there may be details that I'm not totally familiar with, but I thought it would be good to have some biological context for some of the things we're going to talk about, because we haven't really gone into some of the details of that. And so you'll see some slides that have a different format and say, courtesy of Sam Heaps, which are the slides that he prepared for last semester. We'll continue with a discussion of within host viral replication models. We'll try to add cell death. We'll try to add the immune response. And then we'll talk a little bit about some principles of modeling, because there's a lot we don't know about how to model the immune response. And I'll talk a little bit about some of the ideas about how you generically can make those models. And then I don't think we'll get to it today, but Josh has some code to do fitting to the experimental data, to fit this model to the experimental data to see how well we do. So that was the plan. I wanted to go over the homeworks from last time a little bit. I thought people generally did fine. That some of the questions are rather open-ended and maybe not as clear as they could be. In the first question, you were reading the paper. Ironically, the paper that we asked you to read was the one we didn't replicate rather than the ones we did replicate. And the question was, what was it trying to answer? And then what are key assumptions the paper makes about RCV? And then the main mechanisms it's trying to investigate. And I think the word mechanisms maybe wasn't so clear. That'll come up a lot. The word mechanism, sometimes the word process is used. And what I was trying to get at there was essentially what were the biological functions or processes that were included in their model. And their base model actually looked a lot like the model we're developing here, where you have a virus, the virus infects cells, those cells then, those individuals go through early infected, late infected stage. They go on to infect other individuals. It had a birth process for new births. It had an aging process where people who were initially in the 0 to 1 year group moved into year 1 to older group. It had death processes where people could die from the virus or otherwise. And it also had the idea that you could be immune to the virus for some amount of time, but you could potentially stop being immune to it. And so those were, when I was talking about mechanisms, what I meant about that. And then the second question was, what do the subpopulations represent? And they were age stratified populations, 0 to 1 year and then older. And the question was, why is that? And are those choices reasonable? And the key thing here is the assertion in the paper, which one could accept, although I guess one could validate it separately, which is that infants under the age of 1 year are much more likely to have severe infection. They get the infection more commonly and they're much more likely to be hospitalized. And therefore, if you were looking for severe cases, it made more sense to address them as a separate population. And that they had data on whether they were infants or above 1 year, but the base data set they were working with apparently didn't stratify by age above 1 year. It's a little surprising, but that's what the paper claims, I believe. And then the question about seasonal variation in infectivity, and is that reasonable? Well, there's certainly been a lot of talk in the context of COVID, and certainly with influenza, that outdoor temperatures and behavior changes as temperatures change could lead to effectively different R naughts as a function of the weather. When it's cold, perhaps the virus survives longer in the atmosphere, so it's easier to spread by aerosol routes. Perhaps people are more packed in together inside and therefore they are exposed to each other more. But the key thing was that there's believed to be, for some viruses, a weather-related component. They used a sinusoidal variation of infectivity. It's not obvious that the weather is sinusoidal, and therefore the degree to which that's a good approximation isn't so obvious. But as far as their very simple model went, it wasn't unreasonable. But the key thing was that it was basically trying to match the weather to infectivity. And then in terms of the public policy, was that, what were they trying to do? Well, if you read they don't do anything very sophisticated. They say that each vaccination costs a fixed amount. And therefore, the more people you vaccinate, the more expensive it is. They assume that each person who gets sick costs a certain amount in terms of the time in the hospital. Being in the hospital is expensive. Or if they're not quite so sick and they're at home, one of the parents might not be able to work and stay home, and therefore they attribute a cost per day to the lost work. And so there's essentially a cost per infection due to lost work and hospitalization costs, and a cost per vaccination. Those are assumed to be linear in the number of incidents. And then they try to come up with an optimum based on that. I'm not sure whether that's reasonable or not. The cost of hospitalization is maybe not so obvious because in principle, the hospital costs money whether there's somebody in it or not. And so it's not clear how much excess cost you have when somebody's actually in the hospital, unless you have to increase the capacity of the hospital. But certainly if people are not working, there's a cost. It's probably hard to estimate that exact cost. And the cost of vaccination also may be complicated because reaching X people may, or 2X may cost, it may not be linear. It may be quite expensive to have any vaccination, to roll out a vaccination program. Increasing the uptake some may not be very expensive, and then trying to reach people who are very hard to reach may be very expensive. So it's not obvious that those things are linear. But certainly the basic idea that you do a cost-benefit analysis is reasonable. So those were some of the issues that came up in that first question that one might have addressed. And some people addressed all of those issues in one way or the other, but I wanted to go over some of those issues. The second one was the parameter estimation. And the key question here was picking windows. If you look at the curves, both the upside of the curve and the downside of the curve on a semi-log plot are not straight lines. The slope of the semi-log plot changes, which means the exponent changes, which means your estimates change. And therefore you have to think a little bit about which part of the estimate is the one you think is most reasonable. If you're going to go back later in the last problem and actually do a fit, then it doesn't really matter whether our initial estimates are perfect or not. If you're just going to do it by eye, then it matters. And the question here is, very early in the infection, they may not be very accurate measurements. Later on in infection, it may not be a pure exponential growth anymore because other things are kicking in, like changes in behavior. And so you could ask the question, is the earliest phase of the infection the one that's going to tell you what the R naught is for later phases? And on the clearance side, we'll come back to that in a minute. Now, we set ourselves a little bit of a trap. We're going to have two places where we set ourselves a little bit of a trap that I want to come back to. And I asked people to put their results into that table, which was great. But it would have been nice if you were doing your write-ups to actually type the numbers and make a few comments about it. Because in general, people found that New Zealand R naughts were much smaller than the R naughts for Italy and Spain. And so it would have been interesting to come up with a number for that. And the clearance times, the recovery rates were much faster for New Zealand. And so commenting on that and comparing that in the text of your response might have been helpful. I certainly didn't take any points off for not doing it, but if one were going to develop those ideas, that would be nice. Question 3 was fitting the data. Now, this is one of the places we set a trap. So initially, when we did this, we fit the new cases data. And the problem with the new cases data is it doesn't really tell you anything about directly about the recovery time. The assumption in a case like Italy or Spain from the data sets we had was that there were no changes of behavior up to the time there was a lockdown, and then there were essentially no new infections after the lockdown started. For Italy, that may not be too bad an assumption. For New Zealand, it may not be too bad an assumption. I don't know about Spain. But we then switched to the active cases data set, which in principle shows you the upside and the downside of what's going on. The problem is if you actually look into how the active cases are counted, the concept is fairly nebulous. And actually, I think it's on the next slide rather than this slide. If we had a data on the number of people in the hospital, that would be more useful because generally when people enter the hospital, it's recorded. When people leave the hospital, it's recorded. But if you're diagnosed with the disease and you're not under intense medical supervision or you're not hospitalized, how would anybody know when you got over the disease or not? And how would that be defined? And so you certainly would expect that while the R naught might vary between countries, the recovery rate should be the same, though. The length of time you're sick should be independent of the country. And so the fact that our recovery rates differed so much suggests that the way that recovery was estimated in the different countries is different. And I'll show you, I guess, in the next slide where we come back to that and why that may be. The number of susceptibles is also a rather complicated concept. In principle, everybody in the country is susceptible. In practice, the estimates for that S0 that you got were orders of magnitude lower than the total population. And it's worth thinking about that. If the population is very spatially isolated, like the only people who get sick are in New York, then your S0 would be the people in New York, not all the people in the US. But one would like to understand why the S0 is so much smaller than the total population of the country and possibly look at the ratio of S0 to the total population country by country. And that gives you a pretty good estimate of how effective the country was at preventing spread from localized transmission to global community transmission. And that actually could be something one could think about. So here's the definition from Worldometer of active cases: total cases minus total deaths minus recovered. That seems seems straightforward, except that, as it says, recoveries is highly imperfect because reporting can be missing, incomplete, incorrect, based on different definitions or dated, ... And so if you notice, WHO recommends the following criteria: symptoms resolve, that's okay. That means that somebody actually observed that you got better, plus 2 negative tests, that's great. But most people never are tested that way. Symptoms resolve plus 14 additional days, which means we're prejudging how long the recovery is going to take. Can't be faster than 14 days. That would assume if you got sick and recovered in 1 day, it would be 15 days. So you can't possibly get a number less than 2 or 3 weeks out of that. And then this is only a recommendation. Discharge from hospital is all right, maybe. But then some health officials now consider anyone who's diagnosed with COVID 3 or more weeks ago has not died to be recovered. So that means that that curve is calculated by assuming that everybody recovers after 3 weeks. In which case, it's not surprising that our recovery rate was once per 3 weeks, because that was assumed in the data set. So our fit was actually pulling out a number that was already in the data. And so then maybe we should go back to looking at the incidence rate rather than the active cases rate. Again, if one actually has the data, then active cases are really useful, particularly if we had the rate of hospitalizations. That would be really useful, but we don't. And so that was actually a flaw in the way we presented the results and what to do. What we did with it was fine, but the data set we were basing our analysis on was a little bit strange. And then I asked people to vary, once you estimated the rates, to vary S naught, R naught, and mu, and see what happened. And nobody plotted the results. People described the results, but nobody actually showed them. And it would have been really helpful if people had shown some different time series and compared the results explicitly. You could just you can right click on a figure in Jupyter Notebook and save it as an image or as a data set. So that would have been very helpful. And then there was a question about the fit, which was when during the epidemic did the model fit well and not. And actually, Josh was going to give us some pictures for this, so maybe you can paste them into the slide deck for later for upload. But we talked a little bit about this. In general, early in the infection, the fit isn't very good because we don't really know how many people are involved, and that effective S naught is going to change. Then there's a shift in the infectivity because people start being more careful once the epidemic gets going. And so R naught may be shifting. Near the peak of the epidemic, there tends to be a problem, both in the data estimates and in the model fit. And then at the very end, our simulations would say that the rate of infection would go to 0, number of active cases would go to 0. And we don't see that. There's always some, except maybe in China, there's always some residual level of infection because there wasn't perfect isolation. And so essentially, the very early time, very near the maximum, and very late times, the data sets don't fit the model very well. Those intermediate times going up and down, the fits are pretty good. That varies a little bit between countries, and also because of the way the data are reported. If you look at the data in detail, and Josh did some smoothing and things like that. If you look at reported incidence data, for example, in Worldometer, it has a very strict weekly periodicity because reporting tends to be bunched because people don't report on weekends. And so you get higher rates on Fridays and Mondays and very low rates on Saturday and Sunday. I'm not quite sure why there are variations during the week, why Monday, Tuesday, and Wednesday have periodic variations. But within a week, you can't really tell much because there's a huge variation in reporting. And there also are problems. For example, a country will have a bunch of cases that weren't reported at the right time and that all get bunched together. So suddenly 20,000 cases get dumped in on some day. And some countries even took some out. England went in and retroactively took out a bunch of cases, made a hole in their data set at one point. And so one does have to be aware that there are these fluctuations due to human results. I remember a talk given many years ago about people who were observing animals, and they saw some weekly periodicity in animal behavior in the wild. And of course, they said, why is that, to the audience? And the answer, of course, is the animals have no idea what a week is. They might know about a month because the moon might have something to do with it, but the animals certainly don't know about a week. And what you see something on a weekly basis in a wild population, it means there's a problem with your recording. Either your cameraman or whoever's doing the recording is different on the weekend from the weekdays, or they're not working as many hours, but there's some difference in the human side of the observation, not in the data set. Okay. Problem 4 was on herd immunity. Maybe that problem was so simple it confused people, but very few people actually just calculated the number, which is 1 minus 1 over the R naught you just calculated, and gave me the number back, which was all I was asking for. If you thought that problem was really too simple, then you might look at the uncertainty in your estimate of R naught from your fits, and then see what that does to the vaccination or herd immunity level that's required. But literally, all you had to do for problem 4 was take 1 minus 1 over R naught from problem 3 and give me the number back. And so maybe that was such a simple thing to do that it seemed confusing. Number 5 was a more ambitious problem. I asked people to find a new data set and analyze it. And almost nobody did that. People tend to reanalyze the data sets we'd already looked at. It would have been really nice to look at China, New York City, South Korea, Taiwan. Some of those data sets, a couple of people said they looked and couldn't find the data sets. Worldometer is a bit of an annoyance that way, because while you can read off the data day by day by putting your cursor over the graph, as far as I can tell, it doesn't allow you to download a CSV file with the data in it, which is a bit annoying. However, if you do a search, Johns Hopkins has the data sets, and also there's a British organization, ourworldandata.org/coronavirus, which comes up immediately if you search for coronavirus incidence data, downloadable. That's the first item that comes up. And if you click on that link, you'll find that they have a huge CSV file, which has daily numbers for every country in the world plus. And then one can cut and paste the numbers for whatever country you want. They don't really have good data on, they have incidence data. They don't really have the recovery data so much. Some of them do, some don't, because of this issue about recovery being an estimated parameter. But the data sets are pretty available. And it would have been sort of interesting, I think, to compare places that lock down very strictly, like China or New York or Korea, to a place like Sweden or the USA, and ask the question, does the model work equally well for both? Is R naught the same or different from both? And so those were possibilities. Again, I didn't take points off for that, but I'm giving you some ideas about things one might want to do. If you're doing a project, a semester project on this, I'd expect people to dig into these problems more. So that's really why I'm discussing these now. Are there any questions about that so far? Again, I thought people did well. I'm not trying to be critical of what you did, but thinking about how to go a little further with some of these problems. This week, the main homework is going to be, and I think I noted this in everybody, I really would like everybody next week to have some simple simulation coded up in Tellurium of some aspect of the problem in your project. So that means that if you're going to do a cancer model and you've got a paper, you code up one of the ODs and you demonstrate it to your classmates next week. I'm not expecting a full set of slides explaining the model. I'm not expecting parameter exploration, but I would like everybody to have some working demonstration of at least the minimal model that they're planning on using for their project next week to show to the class.
Hey, Professor, I had a question about this week's homework due tonight as well.
Yep.
So for the first problem, when we were comparing stochastic models with your ODE models, I basically am scanning through the R naught values, and then I run stochastic and ODE 50 times, and then compare the maximum number of recovered that I get between the two of them to see that if there's such a difference. But every time I do that, it seems like both models like have the same number of total infections.
So what you're saying is you average the result. The ODE model should give you the same thing every time because it's deterministic. For different R naughts, you'll get different results, but for each time you run the same parameter set, you should get identical results for the ODE model. The stochastic model, each time you run it, will give you different results. And the question there really is the average of the stochastic models the same as the ODE model? Does it converge? So if I flip a coin, I expect that the average number of heads will be half. But each time I flip it, I'll either get a head or a tail, and I'll get some distribution of heads versus tails that will fluctuate. But if I flip it enough times, I expect that counting error, the divergence will go like 1 over square root of n, where n is the number of times I flip it. And if I flipped it an infinite number of times, I should get a half as my average number of flips.
But it seems like every time I run like a stochastic model and an ODE model next to each other with the same,
Yes.
they end up getting the same number of total recovered individuals.
So is it always everybody recovers, or is it that
No,
that
it's always like if 100 people recover in the ODE model, 100 people recover in the stochastic model, even though the rates kind of differ.
Right. So during the simulation they fluctuate, but at the end they come up to the same number.
Yeah,
that's very interesting. So that means that the final number of people who get it is rather independent of the stochasticity.
Right.
But the detailed time series is the same. That's a very interesting result.
Yeah, I'm assuming I'm doing something wrong, but I don't really know what.
Well, I mean, if you want to screen share it, we can look at it together, or we could do that offline. Either way, I mean, if you're seeing fluctuations, and the stochastic model really is jagged, and each time you run it, it's different, but the endpoint is the same, then I believe you're probably doing it right, and it's a property of the model. Some models do self-average in strong ways. Some models give you totally different results. So a classic example of a model that gives you totally different results is there's a molecular circuit called toggle, which occurs in a lot of developmental biology, where gene A inhibits gene B and gene B inhibits gene A. So you either have B turned on and A off, or A turned on and B off. In an ODE model, for any given initial condition, you either wind up with A turned on and B off, or B turned on and A off. That's it. In a stochastic model, you get A on and B off, or B on and A off, but every now and then they swap. And so in time, they sit with A high and B low, and then there are some fluctuations. They both sit near a half, and then they flip.
Right.
And then after a while, they come back. That only happens in the stochastic model. And so that's a case where the stochastic model gives you a temporal result that's completely different from the deterministic one. In other ones, as we saw I showed when we did it in class, if we had large numbers of individuals, the stochastic models varied a little bit from the OD, but not very much. It was only in the cases where we had very few individuals that there was a significant difference.
Right.
And so I have to say, I don't know for the particular case of the final number of recovereds, whether I would expect it to be exactly the same or not. Does anybody else have an observation on that, either agreeing with Antonio or disagreeing with it? That's a profound question. I wouldn't assume that you did it wrong.
I can share my code if you'd like.
I mean, do you want to screen share it now, or do you want to do it offline? Either way, you tell
Either
me.
way, I'm happy to do it either way.
I mean, I can certainly stop sharing for a second if you want to show us what you got.
Sure.
You got it running.
So here's the code. Here's where I'm scanning through R naught via gamma. This is just a counting term. And then I'm running the simulation 50 times. Each time I'm making sure the change to gamma, I'm doing my ODE simulation and my stochastic simulation.
Right.
Finding the maximum R values. And then, if the maximum value is, less than 15% that of the ODE, take that counter, calculate the R naught, and then plot the percentage of
Cool.
percentage
Okay, so
eliminated.
right. So this was the extermination result. Okay. I thought, okay, I wasn't sure. So here, and so what you got here was that nothing happened. Is that right?
Yeah. So if I print out like the maximum values
Right.
of each gamma set,
Hmm.
they're always the same. So the first number is going to be your stochastic maximum.
Right.
And the second one is going to be your ODE maximum. But for each gamma, they're exactly the same.
Hmm. Okay. So can we do? No, I get it.
Oh, I guess they start getting a little larger.
Yep.
But then,
Right. So I think the problem is if R naught is very small, nothing happens. And so when R naught is small, essentially you have a trivial result where everything is boring. So one thing you could do, and then let's just do this once, and then if anybody else has suggestions, but we'd probably do the rest of this online. But let's go back to your code for a second. And instead of doing the simulation, doing 50 replicas, let's just do 2. And let's actually plot the time series. So do a after you've done the O equals M dot simulate, do a inside the loop, do a R dot plot or an M dot plot. And then do the same thing after the Gillespie. And let's run that and see what happens. Why is it not?
oh, is it because
Okay, because you don't have the time. So you'd have to put the time into.
I think this is going to throw an error.
Yeah, you may have to.
I think I have to adjust.
You may have a trouble with 40 and 40. Let's try it and then we'll see. 40 and 41. So there, okay, so R. So R is doing recovered is all right. And the second one is giving us a constant. So you're not getting any infections in the second one. So I think I think we'll have to look into that a little bit more to see what's going on. But I think what I'd do to debug that would be to actually look at the time series and see if the time series are reflecting the calculation that you're getting for that argmax.
Okay.
Because I think somehow maybe either the data set isn't quite what we think it is, or that argmax isn't doing the calculation that you expect.
Okay.
Again, I don't know if anybody else did. Anybody else have either success or failure with that homework problem?
Callie seems to have mentioned something in the chat.
No, I was just asking what kind of model we were supposed to run. I'd assumed an SIR model for the first problem, and I think that was right.
So you also had something where you got a trivial result.
Or did you gotten quite that far yet? So no, it was just a question about the type of model to use.
Right. Right. No, I expected you to use the same SIR model we did in class, the same one. There was a slide where I had the extinction rate plotted from that source. And so it was the model on that page. That was the one I thought we would do. And I when I assigned that, I was a little bit nervous about whether that might be more difficult than it was intended to be. If it was, I appreciate your patience with it. And I'll certainly make allowances for that. One of the problems is how you would define extinction. But which is why I used that tolerance metric for it. The source that I looked at didn't actually go into detail about how he did the calculation. And so that figure was a bit of a mystery. It came from a slide deck from a class on SIR modeling. And so we could look up in more detail what the paper was. But I do recognize that was a little bit trickier as a problem because you had to get an estimate of first you had to get an estimate of what you expected, which was from the ODE model, and then you had to look in the stochastic models and see what happened. Conceptually, extinction is easy, but in practice, it may be more difficult. And so if people found that, as I always say at the heading of the homeworks, if the problem seems unreasonably difficult, tell me, and we'll try to make allowances for it and go over it. So now that I know there's an issue for next week, Josh and I can look at that in more detail and see what we'll come back and try at the beginning of class to come back and have an explanation for you.
Thank you.
Okay. And thank you for digging in and trying it. I appreciate that. Unfortunately, since this is a new class, I haven't given these assignments before. And so sometimes they're not quite what we expect. Sometimes there's a typo in the assignment, which makes it harder or easier. Sometimes it's simply that there's a problem that seemed easy when we worked with it, but was actually had something in it that was a little more difficult than it was intended to be. That one was one that we added a little bit at the last minute, so potentially was less debugged than it might. So that was what I wanted to say about the homework. Again, the assignment for next week, we may assign one or two small things, but the main thing is I'd like people to dig in and try to get some tellurium running based on one of the ODE models you've been looking at and be ready to show that to your classmates. You don't have to you could have one slide if you want, or two slides, but you don't have to. I would like you to show the notebook and show what it does and maybe say a few words about it. And the reason for that really is just to make sure that everybody is actually digging in and having some results. I know many people are, but I don't want anybody to be left behind on this. And so I guess you could think of that as your midterm if you want, is to have some small result. And it doesn't have to be fantastic, but it should run and it should display something. And it should be based on one or more of the papers you've been reading. And I'm happy to talk to you individually about your particular cases. So now we're going to come back to the in-host viral dynamics. And we had started with this nice review article of Amber Smith, Validated Models of Immune Response to Virus Infection. And she had this simple diagram of virus getting into a potentially infectable cell, that cell becoming an I1 infected cell, early infected cell. Once the cell has produced enough virus, releasing the virus into the environment, the infected cells can die. In this case, the susceptible cells can recover, although we are not going to include that in detail in the model we've been addressing. In uninfected cells could die too. In epithelial tissues in particular, cells have finite lifetimes. They have a natural turnover. So that's a little bit like these SIR models with birth and death, which we didn't really get into as much as we could have. And then she showed some different shapes of viral loads. what she called monophasic, biphasic, triphasic. I'm not exactly sure why these are both labeled biphasic. I think that's probably a mistake in the figure. But in any case, this is a persistent. This last one is a persistent infection. This one is an infection that clears. And then we compared that to some data, in vitro data, for COVID in some cells that are not normally the host cells for COVID. The cells that normally get infected, at least to begin with, are cells in your nose, your throat, your lungs. The cells here, Vero and HUH, are not lung cells or vomeronasal cells. And so it's not obvious why they should be a good target, except that they're standard cells that are used, cell lines that are used for studying viral infection. And in that case, we saw there was a very big difference between the the production of virus from the two cell types. The other thing that was interesting here, which we didn't talk about so much, was that they measured the amount of virus in the cell and also the amount of virus outside the cell. And that gives you a sense of the time that it takes to go from these I1 cells, where the virus is replicating in the cell but not released, to these I2 cells, where the cell virus is actually being released by the cells. And that gives you a sense of how long that delay is. And in those green cells, it takes a very long time. The cells start producing virus inside, but it takes a very long time for any of it to get out because they don't have the mechanism to let the virus out. So those cells would be significantly less infectious than the others. Notice all of these time delays are on the order of 6 to 12 hours. One of the problems when we look at viral data sets is that usually the measurements are made every 12 hours or every 24 hours, and a lot's happening on the time scale of a few hours. And so you miss a lot of potential information because the data sets are not collected with fast enough resolution. So we've been working with this data set that was collected by Amber Smith using from this paper of hers, influenza virus infection model with density dependent supports biphasic viral decay. It's a very pretty data set, a lot of replicas. It's in mice. It's on influenza. The frequency of data collection is once per day. So you might like to have more frequent data sets, especially early on. Reminding you that this TCID number is an operational definition of the amount of virus. It's essentially how infective the supernatant over the virus is, over the tissue is. It makes sense to do that because if you counted individual virus particles, you don't know whether they're infectious or not. Potentially only a small fraction of them are. But it also means that you can't correlate it easily with other measurements of the virus. And so there's a bit of an unknown, which is how you convert that TCID 50 number into other things, like how many RNAs you have of the virus, how many viral capsids you have, and so on. Those are unknown. It's a little bit like that S0 number for the epidemics, which is there's an effective population that could be infected, but we don't know where it comes from. And so that TCID number is something you can measure experimentally, which is nice, but relating it to how many viruses each cell produces is not so easy. And if you look at the talks given by Alan Perlson, there's a difference of sometimes 300 in his estimates of those numbers. It's a pretty big deviation. We've identified the phases of infection, this lag phase, this rapid exponential growth, saturation, exponential decay at a slower rate, and then rapid clearance. I hope I was consistent, I may have missed some. I rewrote all of the slides to put it in units of days. So before we had hours for some of them. So I tried to be consistent now in how I was quoting things as tenfold per X days. to be consistent. And now I wanted to talk a little bit about viral infection. This is a slide that should have been really much earlier at the beginning, but I'm adding it now because I'm coming back, which is that when you're going to become infected in the first place, these are now slides with Sam. You'll notice I've tried to keep the format separate so you know which slides come from Sam, so it's clear. Sam's a great guy. I was really impressed last semester when he gave these lectures as a master's student. He gave 2 lectures for my class. They're really good. They're recorded. If you want to hear his lectures, you can do that. He probably will give a better version of the slides than I will, to be honest. I may interpret them differently. So any mistakes are my mistakes. But one of the things that has to happen if you're going to become infected at all is that the virus has to get in and replicate. And there are a variety of stages in your body, some of which we've talked about, which try to prevent viral infection. There are studies, for example, in seawater, looking at a milliliter of seawater and trying to count the number of viruses. And the number of different strains of virus and the number of individual viruses in 1 little milliliter of seawater is amazingly large, really amazingly large. And the bacteria and microbes that live in seawater are fighting a continual battle with viruses and actually have relatively short lifetimes because there's so many viruses in seawater. That doesn't mean it's unsafe for you to swim, maybe because of bacterial pollution, but those viruses are very highly adapted to their specific hosts, which are unicellular organisms. They don't typically cause humans any problem. But we're surrounded by viruses all the time. And I mentioned that, for example, our skin has RNases, molecules that break down the RNA. And that's an antiviral defense. There are waxes and other molecules or species in your sweat, in your skin, in your lungs, in your eyes, that are all there to keep viruses and bacteria from getting in. And there are also mechanical barriers like the skin, the dead layer on the top surface of your skin that are there. And so to get a virus into you to become infected, you have to cross those barriers. And depending on the particular tissue, whether it's oral, nasal, skin, eye, GI tract, lungs, rectal, urinary, they're different issues. They're all places of viral entry of one sort or another. Then there's what's listed here as intrinsic immunity, which are non-cellular responses. Some of those are biochemical, like those RNases, again, to some extent. Some of them we'll talk about in a little more detail. Then once you get past that, there are cellular innate immune system responses. We won't really talk about that in the context today, and maybe 1 or 2 slides. And then once you're actually really infected, which is the model that we've been talking about, then you have acquired immunity. And we'll talk a little bit about that. Really, once you get past this wall here, you are infected. And so our models assume that whatever has happened, some cells have become infected. And so we're not looking at those barrier functions, but it's something to think about. And that's the reason why you can have 2 people, somebody can cough next to 2 people, one of them gets sick and one doesn't. So there is stochasticity. We've talked a little bit about the process of viral infection. We'll come back to this in more detail later today. In general, you start out with viral binding to epithelial cells. That's the process of infection model last week. The cells are going to produce something called a cytokine, which we'll investigate a little bit more. That cytokine will signal to a variety of what are called antigen-presenting cells, here labeled dendritic cells. Those dendritic cells will actually move by crawling to the lymph nodes. where they send signals and do a variety of things that send both cells and eventually antibodies back to the tissue that's infected. And we'll come back to that a little bit more. But the main reason that the adaptive immune response is slow is that it takes time for these dendritic cells to get to the lymph nodes. The cells in the lymph nodes have to mature and multiply, and then they have to get back to the tissue. And that's why you have this typical 10-day, more or less, response time for almost any viral infection. They can last longer, but they can't last shorter than that because they're intrinsic timescales of the immune response. We also, last time, we talked a little bit about the complexity of actual replication of viruses inside of cells. That's a bit of a separate problem. In the case of COVID, you have a single-stranded positive RNA virus, and we talked about that. All of that subtlety and interest of viral replication is ignored in the models that we've written down, except for the fact that there's this I1 phase and I2 phase. But in fact, if one wanted to understand, for example, drugs trying to treat the virus, antivirals, you have to understand the life cycle of the virus inside the cell in more detail. Based on that, we built what we called sort of a base model of viral infection in tissue. And the fundamental concept was that we have some virus, we have some infectable cells we call T. The virus gets into combines with the infectable cells T to make our I1 cells. There's some rate at which the I1 cells become I2 cells, and then they begin producing virus. And we guessed some numbers. The beta we pulled from Amber's paper originally for influenza. The rate at which the I1 cells became I2 cells is 4 per day. Came about from that slope that we saw for the lag phase that we saw in the experimental data. The production rate of TCIDs per cell per day was essentially an arbitrary number. Didn't come from anywhere. Well, we know effectively because the amount of virus we have here doesn't really have an independent meaning. The only thing that really matters is beta times V. That's significant, but the units in which V is measured aren't significant. And then we did a simple model of that. We got a number of uninfected cells coming down, the number of infected cells going up, the number of early infected cells there. And this looked exactly like the SIR models that we did before, basically identical. And we were able, by playing around a little bit with those numbers, to fit the delay that we saw early on in viral infection, the lag phase, and the early exponential growth of the virus using the parameters here. And so that very simple model gave us the first 2 steps of this process. Then I asked you to do something. This was the second thing I did sort of on the spur of the moment and wound up making things a little bit more complicated perhaps than they needed to be, and set a little bit of a trap for myself, which was I said, well, we know that when cells are infected, the virus is used up. The same virus particle can't infect 2 cells. And so what I asked you to do was replace T goes to I1 at a rate beta VT to T plus V goes to I1 at a rate beta VT. And we played with that together, and we found that the viral load curves were very, very different. And essentially what was happening is because we had a small amount of virus and a lot of cells that could be infected, early on, all the virus got used up. And so the amount of virus in circulation went down like a rock. And then there was a long delay, and then things came back. And in principle, that was correct. But what I was not thinking about carefully was that the units that we're measuring virus in are not viral particles. which are the things that are infecting individual cells, but rather TCIDs, which are these sort of funny operational unit. And one TCID is definitely a lot of viral particles, but we have no idea how many. And so instead of saying T plus V goes to I1, where I need to use up 1 unit of virus to infect 1 cell, I should have been saying T plus alpha V goes to I1, where alpha is a small number. And what that small number is not so obvious. And I think, given how the time is going, I'm not going to do this as an exercise, but my original plan was to do a little exercise with you to come back to where we were in class the other day, and explore as you changed alpha. Clearly, if you make alpha very, very tiny, you recover the limit where you're not using up the virus at all. The case we did was alpha equals 1, where it takes 1 unit of one TCID to infect 1 cell. And it would be rather interesting, and in principle something one could, if you had good experimental data, one could get a conversion between the actual number of viruses needed to infect 1 cell and the TCID unit by doing this fit. And that would actually be something quite worthwhile to do, because in general, there isn't any good way of connecting those numbers together. So for today, I think we're going to skip this unless somebody really wants to do it. But it's something that might be worth doing as a little homework exercise, or on your own if you're interested in this problem. If your project is going to involve, for example, in-host viral dynamics, I would suggest that investigating this issue is worthwhile. And I don't think any of the papers we look at that I'm aware of ever do that. They all assume that alpha is so small that you can ignore this. But I think it's an interesting problem. In any case, that was I was doing a bit of tail chasing, I guess, when we worked on that. Principle is not wrong, but that alpha factor is significant. One annoyance you will find when you use tellurium and antimony is that you can easily write T plus 0.01 V goes to I1. Antimony will let you write any what's called stoichiometry you want. But this parameter alpha cannot be a variable in antimony. And so what you can't do, which is what you'd want to do, is write a sweep where you change the value of alpha in a loop outside of antimony and run it and see how it's different. And so it doesn't, as far as I can tell, let you change that parameter directly. What you could do, however, is if you change T and keep alpha constant, effectively what matters is the ratio of T, V, and alpha. And so you can change the initial level of T or the initial level of V, keeping alpha fixed. And that's equivalent to changing alpha. So you have to be a little bit ingenious if you want to do that. But the naive thing, which is to say T plus alpha times V, in the way you type it in antimony won't work. You have to have a little bit of a workaround for that. So that's something to think about. Okay. So now I want to come back to something that we talked about last time computationally, but we didn't really discuss in detail biologically, which is, we said that the extracellular virus has a lifetime. And I mentioned briefly that there are a number of different ways that virus can be cleared from the fluid in your lungs, for example. One of them is that the lungs have cilia, which push the mucus up out of the lungs, so you cough it out. And that physically gets the virus out of the lungs. Another one is that that the virus itself can break down biochemically. So there are molecules in the mucus that break the virus down. So lysis would be an example of that, where there are actually molecules that attack the virus and break it down directly. Another one is that there are molecules that stick to the surface of the virus and lead to the virus being eaten, being phagocytosed by phagocytes, which take it out of circulation. In some cases, the virus can then infect them as a host, which is a problem. So there are actually cells that walk around and scavenge virus. And that scavenging can be mediated. They can do that directly or can be mediated by stuff that sticks to the virus. Another one is that there are non-specific antibodies that can stick to the virus, make the virus glom to each other and become unable to enter cells. And so that's another thing that can happen. So the host already has a variety of components not in the adaptive immune system, but in the innate immune system that are present that serve as sort of generic ways of trying to protect against viruses. If you've been exposed to that virus before or a similar virus, you may also have specific antibodies that bind to that specific virus and make these processes more efficient. But there's some generic mechanisms, biochemical and mechanical mechanisms that exist to clear the virus out of the circulation. Something that Sam's slides talk about, which we didn't talk about a lot last time, is that viruses have evolved, not in the case of COVID because it's not co-evolved with humans, but viruses like influenza that are primary human host have co-evolved with our immune systems. And so they have typically developed molecular strategies to defeat the protective mechanisms that we have. If they hadn't, we wouldn't get sick from them. And so there's always a bit of an arms race between the host immune system and the virus. And so, as this was an example for a particular virus, not one of the ones we're going to talk about, where the virus is fighting back against some of these molecular species that are in the circulation that are acting to prevent the virus from spreading. That issue of the virus and the host in arms race will come back more strongly in some of the other cases we'll consider. It's not going to be in the model that we have here, but it is something that one would think about if you wanted to compare, for example, human response to COVID and bat response to COVID. A lot of the difference will come about because of that. So based on this idea that there are these factors eliminating virus from circulation, we introduced a term V decays, rate CV, C is the clearance rate, which reflects a whole bunch of different mechanisms. It could be complement-induced breakdown of the virus. It could be the natural lifetime of the virus in solution because it has a finite lifetime. It could be the clearance of the virus due to the cilia in the lungs. It could be scavenging of the virus by phagocytes. And so there are a whole host of different mechanisms that could be active that reduce the amount of virus in circulation. The key thing here is that the idea is that the rate of elimination of the virus is linear in the amount of virus. Clearly, if there's no virus, the rate of elimination has to be 0. So at least if the amount of virus is small, that's a reasonable assumption. Whether the clearance rate is still linear in the amount of virus if you have a lot is not so obvious. One thing that this tells us, if you have this kind of what's called first order decay, And if we taught this course from more classical chemical kinetics, that one of the first concepts we look at is the first order decay and saturation. The moment you see a linear decay like that, CV, if you had no supply of virus, that would lead to an exponential decay in the amount of virus. If you have a constant supply of virus, then the equilibrium amount will be the supply rate, P, divided by the decay rate, C. And that'll give you the fixed point. In this case, there isn't a constant supply because it depends on the amount of I2, the number of I2 cells we have. And we had to guess a clearance rate. And for flu, the guess is about 2.5 hours, which is about a tenth of a day. So the clearance rate is 10 per day. Actually, we used 9.4 because 2.5. I don't know why I picked 2.5 instead of 2.4, but I did. No. Using hours and minutes gives you these awkward conversion factors, just the way you probably never actually did any calculations using imperial units. But if you use mass and weight, if you measure things in pounds, the unit of mass, weight is in pounds, the unit of mass is slugs. And you have all sorts of strange conversion factors when you have gallons and gallons, hours, British thermal units. The metric system is much simpler. Here, we have to deal with the fact that some things tend to be measured in hours, minutes, and we're measuring everything in days. And so we have 24s and 60s floating around, which is why it's 9.4 instead of 10. Okay, so we did that together. And when we did that, our viral load curve went up. It didn't change the growth very much, but now we got a flat top saturation, and that saturation at a maximum is what we expect when we see what's called first order kinetics. And it doesn't change the cell population numbers very much. It slows things down a little bit at the end, but not very much. All right. And that, is that where we stopped last time? I think so. Maybe we went one slide further. All right. So the next thing that we want to talk about is the fact that cells die. And during infection, cells die in a variety of ways. They can die because the cell simply is overloaded by virus. Some viruses can only be released from cells by breaking them, physically breaking them and killing them. Those are called lytic viruses. COVID is not a lytic virus. Influenza is not a lytic virus. They are secreted through normal cellular secretory pathways. Nevertheless, infected cells die pretty quickly. They can be killed by the immune system, and we'll come back to that in a few minutes. And one of the reasons that diseases like COVIDs have things called cytokine storms is because of that. And that's not going to be in our model in any detail. Something we should probably put in, but we didn't. The other thing that happens is one of these early responses, which is now part of the innate protective system that cells have, which is that when viruses stick to the cell surface, components of the virus stick to the cell surface, so fragments of the virus stick to the cell surface, or when the virus is internalized, and when it starts reproducing, all of those processes send signals to the cell warning that there's a problem. And there are a variety of signaling pathways which cells respond to. And the cell can respond by doing a lot of things. It can try to prevent the virus from getting into the cell in the first place. It can block the viral replication. It can try to prevent the virus from being released. And it can also cause the cell to die. And so there are a number of different kinds of recognition systems. Some of them are specifically associated with bacteria. Some are associated with DNA viruses. Our COVID case doesn't ever have a DNA phase, so that's not relevant. But these things called PRRs, pattern recognition receptors, are relevant to our RNA virus. Again, I don't expect people to learn a lot of molecular biology, and they're going when you deal with molecular biology, one problem these days is they know a lot of molecular species. And the names of the molecular species sometimes are informative and meaningful, and a lot of times seem to be just random. And so I want you to see these names because you'll run into them. If you don't retain them immediately, that's fine. It's like learning any language. Familiarity helps, so that if you see the name of a particular molecular species repeated a lot, then it may be worth adding it to your vocabulary. If you see it once and never see it again, then it's okay to just say, well, I saw it and I've forgotten it. If you have a photographic memory, great. If you don't worry about it too much. I feel sometimes especially sorry for people who come to biology where English isn't their first language, because the number of new words and the randomness of those words is really quite challenging. If you're dealing with classic things like physiology and anatomy, then it's also helpful to have learned Greek and Latin before you start learning biology. When you're dealing with molecular species names, the names are really pretty random. At least sometimes they're colorful, and that maybe makes them easier to remember. In any case, in the case of recognizing single-stranded RNA viruses, there are two main pathways. They're what are called toll-like receptors and RIG-I receptors. Josh is working on those in his modeling. And the key result is that, which is why I wanted to use this slide, is that when these pattern recognition receptors recognize their target molecules, in this case, single-stranded RNAs, they induce the cell to produce what are called cytokines. And we've already seen the word cytokine a few times. We've heard the word cytokine storm. Cytokines are the key molecular species, of which there are a lot, that regulate the immune system at the systemic level. And we're going to run into a variety of these. We're going to run into interferons, and we're going to run into interleukins, and just be aware that those names will show up, and there are going to be a lot of them, and we're going to typically ignore all of that complexity. But when you get to interleukins, we'll find out that the numbers go up. I don't know how many human interleukins there are. There are a lot. Well, at least up to 12 or 14 are known and relevant. We'll find out that interleukin 2 is one that shows up a lot in the context of cancer biology. Interleukin 6 is particularly important in viral infections. Interleukins 10 and 12 are also important in viral infections. Some of them promote inflammation, that is the immune response, and other activity of the immune system. Some of them damp them down. The particular model we're going to use doesn't have an anti-inflammatory model. That probably is a mistake. In any case, it's the detection. There are going to be multiple ways that these signals are produced, these cytokine signals are produced. But the first way they're produced is through the cell detecting the virus. And so that's the point of this slide. Another thing that happens is that when cells detect that they're infected, they die. And they can die through a variety of different mechanisms. It used to be the thought that there were basically two ways cell died, which were called necrosis and apoptosis. And necrosis essentially is when the cell dies by being burst or otherwise being knocked over the head. Apoptosis is a more orderly process where the cell actually reorganizes itself, breaks down its DNA, packages what is inside the cell, and releases it in a controlled way. Apoptotic mechanisms tend to get turned down a lot in cancer. So they were discovered primarily initially in the context of cancer. It winds up that apoptosis and necrosis are only broad spectrums of ways of dying. And there are many other kinds of cell death which you could run into: pyroptosis, autophagy, and there are many others. The main one that we need to worry about here is that during apoptosis, the cell reorganizes its DNA. It expresses a bunch of molecules called caspases. Those caspases are like RNase. These are typically proteases that break things down. And so essentially you can think of at least part of what's happening is that if the cell notices that it's infected, it decides to kill itself. Once the cell is infected, in general, they can't survive and clear the virus. The cell will say, I'm infected, and I'm going to try to break down the viral components that could go on to infect other cells. That's a bit of an over-exaggeration. It's also an anthropomorphization of what the cells are doing, but functionally it's not far from reality. Again, caspases are also activated in cancer and in other situations. But the caspases do break down both the membrane, the mitochondria, the energy-producing components of the cell, and would lock also the viruses inside of the cell. When a cell lyses, when it dies through necrosis, the contents of the cell are spewed out into the environment, and that actually can be quite toxic to neighboring cells. During necrosis, the dead blebs of the cell typically are surrounded by membrane, and so the cytoplasm of the cell and the RNA and DNA in the cell aren't released directly into the extracellular environment. Those blebs, membrane surrounded blebs, what are called apoptotic bodies here, are picked up by macrophages, and the macrophages process those and become antigen presenting cells. And so one of the things that happens is the virus, the components of the virus get chopped up into pieces by the caspases. They're present in the apoptotic bodies. They're picked up by the macrophages, and then those can be presented to the adaptive immune system later on. But the key thing is that, and there also are macrophages also have a structural function, which is you have a lot of debris when cells die, and the macrophages come in and clear up that debris. And that has to happen if you want the tissue to regrow. You have to get rid of the mess that's left over. If a house gets knocked over by a tornado, somebody has to come in with a bulldozer and clear the space before you can build a new house. And so one of the roles of certain kinds of macrophages is to remove the debris from the dead cells after they've died and leave space for new cells to come in and replace the dead tissue. And so apoptosis can cause, fundamentally when the cell dies, it's not producing any more virus, so it reduces the amount of virus in circulation. It also then signals that are inflammatory, promote the immune system response. And there are, as I mentioned, a lot of different ways that the virus can stimulate cell death. There are cell surface receptors, which we'll talk about a little bit. There are cytoplasmic receptors. Some of those are simply interference. If the virus is using up all the energy budget of the cell and all of the molecular components of the cell runs out of its ability to maintain itself. But there are also very specific signal responses that can lead to adaptive cell death, either through mitochondrial permeabilization or through other mechanisms. If the cells were actually able to sense the virus very quickly after infection, when they're still in the I1 phase and die, then the virus couldn't spread at all, right? Because if the virus never gets out, is never manufactured enough to get out of the cell, it can't go on and infect other cells. And so in principle, these antiviral protection mechanisms through cell death, if they were very effective, you'd never get an infection. There are a couple of different pathways. I don't want to go into too much molecular detail. There are a bunch of cell surface pathways that look for fragments of virus in the environment. These might be viral DNA or RNA floating around, or they could be viral coat proteins. We haven't talked about transmembrane receptors and G proteins and all of those molecular details, but they typically are transmembrane receptors that bind to particular pieces of virus, and they have death signals typically related to caspase 8. If you're a cancer person, caspase 8 shows up a lot, which is why I thought I'd put this slide in. And that activates caspase 3, which is one of the classic apoptosis mechanisms. Actually, caspase 8 is a little more complicated than that. We used to work with a guy who studied caspase 8. One of the things caspase 8 does in the context of cancer is it tells a cell if it's in the right place or not. If caspase 8 is bound to the membrane, it tells the cell that it's on the right extracellular matrix substrate. If the cell comes off the right extracellular matrix substrate and is in the wrong place, caspase 8 is released and goes to activate caspase 3 and promote cell death. So cancer people might have run into cap8. The other ones are a whole variety of molecular species, some of which I don't know well myself, which can do a variety of things. In particular, they can lead to calcium release of the cell. That calcium release could lead to cell death directly. They could also lead to what's called mitochondrial permeabilization. Your energy mitochondria, which produce the ATP of the cell, have their own cell membranes, subcellular membranes. If you make holes in those, the cells die really fast through reactive oxygen species. And so one of the fastest way for a cell to die is to let its mitochondria leak. And so mitochondrial permeabilization is a classic mechanism of rapid cell death. And there are a variety of pathways that do that. In your cells, there's always, due to especially cancer detection, a balance between survival factors and apoptotic factors. And the mitochondria are always sort of on edge about that. So they're primed to allow cells to die very rapidly. If that happens too much, you could get degenerative diseases. If it doesn't happen enough, you could get cancers. In the context of viral infection, these mechanisms are used in slightly different ways. Just as with complement, viruses have co-evolved for viruses that are present in humans or other host species that they've lived with for a long time. They've co-evolved to block these apoptotic mechanisms. If these apoptotic mechanisms are too effective, then the virus can't reproduce. And so if you're exposed to a virus that you're not hasn't co-evolved with you, say a cat virus, that virus might, if the cat sneezes on you, that virus might get into your lungs. It might even get into some cells, but your cells will recognize it and kill themselves before the virus can reproduce, or at least before it's reproduced very much. If a virus is going to cross species and reproduce, it has to be pre-adapted to be able to suppress these protective mechanisms. And there's some examples here of some of the ways that viruses prevent cells from recognizing them and committing suicide. I am certainly not an expert on any of these, but again, for people who are interested in cancer, human papillomavirus, which is associated with certain kinds of cancer, inhibits p53, which is one of the master regulators of apoptosis. And so there are some viruses that are associated with cancer activation that tend to mess with apoptosis pathways in persistent ways rather than in short-term ways. And so that's something. Herpes virus is another one. I like that. Actually, if we look at the list here, many of these, cytomegalovirus, herpes virus, papillomavirus, hepatitis B viruses, these are all persistent viruses that once you're infected, they stay in your body for a long time. Many of them are associated with higher risks of cancer because of their suppression of apoptosis. So that was a little bit maybe more detail than we wanted, trying to explain the idea that when cells are infected with virus, they die. And the naive idea is that the virus is killing the cells. That can happen up to a point, but it's not just that the virus is damaging the cells and killing them. The virus actually can be actively signaling to the cells, or the cells can be detecting the virus and actively killing themselves, because that reduces the rate of replication of the virus. And so one of the main ways that cells protect themselves, or tissues protect themselves at the tissue level from the spread of virus, is by cell death when the cells are infected. And so we're going to model that in a very simple way to begin with, which is that in fact I2 cells, our infected cells, die. And the higher the rate at which our I2 cells die, the less the virus can, less each cell that's infected can produce virus. And that means that effectively the smaller R naught. Because this is essentially I2 goes to D is like our I goes to R in our SIR model. And so the faster that happens, the less infection we get overall. Notice that I don't have a line going from I1 to D directly. If those protective mechanisms I've been talking about were perfectly effective, or even relatively effective, then at least sometimes the ideal situation from the cell's point of view would be that if it's infected, the I1 cell dies before it becomes an I2 cell, before it produces any additional virus. And so one might ask the question, and I think it's actually would be worth maybe doing as a little homework assignment. I didn't have it here. Maybe make a note of it, would be to add an arrow from I1 to D directly and ask the question, what happens when you increase the rate of death directly from I1 to D? The answer will be that the virus spreads much less. Because of the way this model is structured, it can't prevent viral spread completely, but it would reduce it. The key thing that we need to do is think about how long the infected cells, those I2 cells, produce virus. This is, again, very much like the SEIR model or the SIR model. The total number of people, the total number of infections that are not was the number of infections per day from somebody infected times the number of days infected. Remember, R naught was N gamma divided by mu, where mu was the rate of recovery. So the number of new cells infected by I2s will essentially be the rate at which the I2s infect, that's their rate of production of V, divided by the rate at which the I2s become Ds, that's D. And that will give us essentially this rate of viral production, P, divided by the rate of death, D, is equivalent to N gamma divided by mu in the SIR model. And so P divided by D is equivalent to R naught in our base model. And that's actually a critical number, that P divided by D. Does that make any sense? I hope. So we need to have a rough estimate of how long late infected cells survive, how long they're producing virus. For influenza, a guess of about 4 days is probably not way off. So that would mean the clearance rate D would be about 1/4. For SARS-CoV-2, the infected cells seem to produce virus before dying for about 2 days. So that rate D is about 1/2. Now, if you came back to me and told me that the actual numbers were the reverse of that, or a factor of 2 bigger or smaller, that wouldn't shock me. It'd be interesting to look those numbers up in the literature. They're certainly known for influenza. It'll depend on the cell type. It'll depend on whether you're in vivo or in culture. In culture, you can measure it. And Josh has been reanalyzing some experimental data, what are called plaque assays, where they induce, they infect a small patch of cells, and then the infection spreads in vitro in an epithelial layer in a dish, and you get a ring of I1 cells on the outside, which are newly infected but not producing virus, then a ring of I2 cells inside of them that are alive and producing virus, and then in the middle you'll have a circle, a disc of dead cells that have died through these mechanisms. And so you can actually calculate from looking at the speed at which this ring moves out, how long the cells stay in the I1 phase, the I2 phase, and once they're dead, they're permanently dead. They don't undie, but you can get those rates. So actually you can measure those rates in an experiment pretty well. And so Josh could give us those numbers. Okay. So I think that was where I wanted to come back. This death will give us the clearance term that we wanted. Remember that before we had a delay, exponential increase of the virus, and then saturation where the virus stopped growing but didn't go away. But in the influenza, as we saw in the data set, there's this gradual decay. It's actually pretty rapid. It's still exponential, but it's decay. And that decay is coming about, we believe, from the death of the infected cells. This is happening already by day 2, before there's any adaptive immunity. And so this is the innate immune system kicking in and fighting the virus. And so we'll do that together. So that'll be our first demonstration. We're halfway through class, so do you want to take our 5-minute break? And when we come back, why don't we tool up, load our model and start working from there, okay? Anybody have a strong feeling one way or the other? Okay, Brian, thank you. All right, so why don't we take a 5-minute break? We'll start back at 6:40. Josh, are you there or did you have to run off? It's okay if you ran off.
No, I'm here. I'm just having dinner.
Okay, that's fine. People have been very quiet and I've been talking a lot. So If things don't make sense, you can either put it in the chat or just speak up. And I don't know, I mean, people vary a lot in terms of the amount of background they have in biology. So what I just went over may seem either confusing and messy, or things you knew from 10 years ago. In either case, I apologize. I was trying to give you enough basic information that some of the things we're going to talk about make sense, and not go into too much molecular detail. And it's always a bit of a fine line to try to come up with that balance. So let's take our break, and then I'll see you back here at 6:40. Okay, is everybody back?
Yep.
Okay, great. Were people able to fire up NanoHub and load the model that we were working on from yesterday, from last time? So if you scroll down, you'll find that we're here. Infected cell clearance due to viral infection and innate immune response. And so if we look at the model, this model looks like the model we had previously. T goes to I1, rate beta T times V. I1 goes to I2, rate K times I1. Virus is produced at a rate P times I2. Virus is cleared at a rate C times V. And now there's a new term that the I2 cells die at a rate D times I2. And we said that the rate was approximately a quarter per day. Okay. So why doesn't everybody either load the code that you were using yourselves the other day, or you could load the demonstration. If you have your own code, it's probably better to type it yourselves, but it's here too. And if I run that, I see that, all right, let me see what the differences are here. The differences here are, I have to remember what I did. Okay, so the first one here is the influenza case where the clearance time was, for the death rate was 4 days. And here I've changed K. K is the infection rate. I'm actually not sure why, now that I look at this, I'm not sure why I have that there. I'm comparing the results in these graphs, for one where the infectivity was 4 and another one with a lower infectivity. Not the most obvious thing to do. But this first curve is the one that I was invoking here, where you see that I now have a new population, this red curve, which is dead cells. And so instead of the number of I2 cells gumming up and staying high, I now have the die-off of the infected cells and their replacement by the dead cells. If I look at the viral load curve here in blue, what I see is a peak followed by this slow decay. And that's what we were looking for in doing this. So I'd like everybody to do this exercise. And so I guess I'll do a poll and wait until everybody is able to catch up with that. Because we'll need this as the base for what we do going forward. And I also appreciate people's patience. I try not to lecture usually for a block of 90 minutes at a stretch, because I know listening to me talk for a long time is not the most exciting thing in the world. But I did want to do a little bit more biology, and that meant there was a little bit more lecturing than I normally would do. A little fewer exercises and more lecture. If you've done the caught up already, you could try exploring this, which would be changing D to a shorter lifetime. Here we had D was I said here infected cell lifetime for influenza is actually shorter. Here I said 12 hours, in which case D would be 2 per day. Let's try that, see what happens. Instead of making it 2e to the minus 1, we can make it 2e0. And you'll notice that if I do that here, basically my I1 cells turn into I2 cells and then die right away. So my dead cell population comes up very, very fast. And my viral load curve now has a totally different shape. I don't have a biphasic response anymore. My viral load comes up, max to maximum, and then decays rapidly, very rapidly to 0. And so that shorter lifetime for the cells isn't compatible with the other assumptions that I've made if I want to match the experimental viral load curve. You could also try, we said that for COVID, it was about twice, the lifetime was about half of that for influenza. So let's try 4e to the minus 1. When I do that, it doesn't change things very much. The decay curve is a little bit faster, but not by much. Fundamentally, the slope of this decaying line is fixed by that lifetime of the infected cells. Okay. Does anybody need more time? Is everybody able to do that now? There was one person who had asked for more time. Okay. So this was showing the results, which you've just talked about. Now you see this decay curve matches the experimental decay curve here. And so now we have region 1, region 2, region 3, and region 4 all correctly reproduced. Whether the model is actually reproducing them because it's the right biology is an interesting question. But we're able to reproduce those first stages of the viral load curve. What we're going to do from here on out is going to be dealing with the adaptive immune system, and things are going to get more problematic. And they're going to get more problematic really fast because we have less ability to monitor the adaptive immune system. And the adaptive immune system responses are more complicated. And so we're going to be shooting in the dark a little bit more. In these early stages in this base model, fact that cells become infected, that it takes time for the cells to become virus releasing, that they release virus, and that they die. Those are all relatively simple experimental observations. Those are all observations you can make in vitro. You don't need to be in a living animal to do it. You can do that in a dish. When we get to the adaptive immune system and cellular immune response, you can't do it in a dish anymore. And so making measurements becomes more difficult, more expensive, and interpreting them becomes more difficult. And so now we're going to come back to this picture that we started out with, which is we've been modeling this early phase of infection here. And now we're going to have to deal with some of the complexity of the immune system. And the first thing that we're going to have to deal with in the immune system is the fact that the cells that are going to come in and attack the virus in the tissue, some of them start out in the tissue, but most of them start out in your lymph nodes. And that means that we have to have conceptually a separate compartment, which is going to be the lymph node, where those cells are generated. And that means that our model is going to become a little bit more complicated. The second thing is that we have two things being transmitted from the tissue to the lymph node, one of which are cytokines. These are these chemicals that are released by the infected cells and that get into the lymph and the bloodstream and circulate, including to the lymph nodes. And the second thing is the what are called antigen presenting cells or dendritic cells here that physically have to crawl from the infected tissue to the lymph nodes. And I don't know anyone who's had, for example, a sore throat, but if you ever went to the doctor for a sore throat, you would feel your throat here and say you have swollen lymph nodes. One of the early names for AIDS or HIV infection was related to lymph nodes because you had degeneration of lymph nodes in late stages of HIV. So your lymph nodes are primary responder to infection, cellular responder to infection. And one of the things they do is swell up because there's a lot of cell reproduction going on when you have viral infection. And so when somebody feels your lymph nodes to see if you have swollen lymph nodes, that's checking essentially if you have a lymphatic cellular response, an adaptive immune response. The lymph nodes produce a number of cell types which are returned to the tissue. Some of that is returned via the circulatory system through blood. Some of it's returned via the lymphatic system. Some of those are specific antibodies to the virus. That's a little later. Another important one is what are called cytotoxic T cells, CTLs. And there are a number of different kinds of those. And there are some other cell types that we're not going to talk about so much. Memory T cells, B cells, and a variety of other cell types. But this flow of cells and information from the infected tissue to the lymph nodes and back is something that we have to deal with. And that's a level of complexity that we have to deal with. The particular model that we're going to build is not the most attractive or accurate way of representing this, but it will at least convey some of the ideas that we need to address. And we'll come back reading some papers that deal with this in more detail and come back to it. If we dig into the adaptive immune response a little bit more, we could go into the immune system at some levels is mapped quite well in terms of the numbers of cell types, the molecular species, the locale where things happen. It's pretty complicated. In some ways, it's very poorly understood. why some viruses create strong antibody responses, why some viruses create strong T cell responses, why some viruses don't, why some vaccines, which work by stimulating the adaptive immune system, do and do not create long-lasting immunity, why immunity lasts a long time or not for different viruses. None of these things are very well understood. One of the big mysteries, perhaps the single biggest worry with COVID is that we still don't know how long, if you're infected with COVID and you survive it, how long you're immune to it, if you are. There's some viruses, again, that we talked about earlier, where infection once in your life essentially gives you lifelong immunity. There are other viruses where you can be reinfected a week later. There are other viruses that seem to reset the immune system so that you lose the immunity that you had to everything else. So exactly how and why those things happen is not terribly well understood. And certainly if we don't understand it, we can't model it. Models aren't magic. If we were going to dig into the T cells a little bit more, the T cells have rather a complicated life cycle. In this diagram, they're shown as all happening synchronously at some level. Some of these things happen at different developmental stages. So one of the big problems with the immune system, both with antibodies and with T cells, is that you need your T cells to come in and kill cells that are infected, but you also need them not to kill cells that are not infected. In the case of cancer, you want them to come in and kill cancer cells and not normal cells. And so non-self recognition is a critical problem. If you have T cells that come in and kill cells that they shouldn't, you die of autoimmune diseases, or at least you get very sick from autoimmune diseases. And we have plenty of autoimmune diseases like arthritis. So this is something that we all suffer from, especially as we age. On the other hand, if they don't kill cells that they should be killing, cancers can develop and get away, and infections can spread unchecked. And so how the immune system generates T cells that recognize invading pathogens and don't kill normal cells or uninfected cells is been pretty problematic. Some of that is understood better now, and some of it has to do perhaps with why COVID is more lethal in older people than in younger people. So T cells have a very complicated, the cells that do the adaptive immune killing, has a very complicated life cycle. They start out initially in bone marrow. They then go to the organ called the thymus. And the thymus, and there are two main varieties of there are quite a few varieties of T cell, but the two of the ones that we're going to care about are what are called CD4 plus and CD8 plus T cells, which are associated, we're not going to model those today explicitly, but they're associated with particular molecules on the surface of the cell. Both of these cells have particular immune-related, antibody-type immune-related receptors that are highly variable that recognize targets. And so these are actually due to DNA shuffling that change so that we tend to think that all the cells in your body have the same DNA. Immune cells actually have different DNA from other cells in your body, and each immune cell of different type has different DNA sequence. So there are DNA regions that are highly variable that generate the combinatorics to allow for the recognition. How do you recognize something that you've never seen before? You can't know it in advance. So what your body does is generate all sorts of different things that recognize all sorts of different things and hope that one of them will recognize the pathogen. If you're unlucky, none of them do. If you're unlucky, some of them recognize self cells as pathogens. So the initial cells are generated in the bone marrow, and then they go to the thymus. We're not going to model the thymus, although we've talked to people who are trying to do that. And the role of the thymus is to do two things. Its main role is to make sure that cells that recognize self get filtered out. So the bone marrow generates cells with random recognition, and a lot of those cells would recognize self tissue like heart cells, bone, cartilage, neurons. And if those T cells then became activated, they'd happily go around killing those cells that you need. And so the thymus has a filter that says it takes these what are called naive T cells and says if those T cells bind to cells in the thymus, then they're killed. And that filtering mechanism has to be very, very good, because even a few T cells that go rogue can cause very serious disease or death. On the other hand, if the thymus filters out too many T cells, then when you're infected, you won't have the variety of response that you need to be able to deal with the infection. One of the things that is interesting is that as you age, your thymus shrinks radically. In your 20s, you still have pretty good thymus. By the time you're my age, the thymus is almost gone. And that means that your body's ability to generate new responses to pathogens decreases radically as you get older. And so that may be one of the reasons that older people have more difficulty dealing with new infections. If you're exposed young, then you select for those T cells young. But if you're older and you didn't have the T cells in your repertoire, you're not able to generate new T cells. So that's a problem. The T cells, after they've gotten through the thymus filter, then go to the lymph nodes, and they sit there. And they sit there until one of these antigen-presenting cells, those dendritic cells, crawls to the lymph node. The T cell that antigen-presenting cell will present fragments of the virus to all of the T cells randomly. The T cells have random receptors on them. If by accident one of the T cells has a receptor that happens to glom onto the virus, then that will send a signal to that T cell to activate and start replicating. And that sounds reasonably complicated, and I don't really have any way of simplifying it. I don't think there is a simpler way to explain what happens. Now, there also are a second set of responses which simulate what are called B cells that produce specific antibodies to the virus. And we're not going to go into that today, and probably not much in this class, although it's a very important set of questions. We're going to focus on the cellular, the T cell effector T cell response. But similarly, the B cells are sitting there waiting for a signal. And if they get the right signal, then they grow and they release antibodies, targeted antibodies. And so you have both things going on. Exactly why you have to have CD4 cells and CD8 T cells is essentially a security mechanism to try to inhibit autoimmune response in tissues. It basically is there to limit the CD8 plus T cells from going rogue and killing too many target cells and non-target cells. We're not going to, in this class, model CD4 plus effect, although it's important. Okay, any questions about that? Have people walked through the immune system before? How many people have seen a basic presentation of the adaptive immune system before? Colleague. So, okay, so this is review for you. If it's, okay, good. So it's review for most of you. In that case, I will ask you, if I say something that's wrong, please speak up, because you may very well know as much or more about the immune system than I do. And so if I misspeak, don't be embarrassed to say, hey, you said the wrong thing, or you missed something. Because the immune system is not my area of expertise. And so you may very well know details about it that I don't know, or I may make a mistake. But this is the basic idea of the adaptive immune system that we're going to. Okay. So the key thing that we're going to be talking about are T cells. And what we want to do is have a model of T cell production. And we're not going to worry about bone marrow and thymus. We're going to assume that there are quiescent T cells in the lymph nodes that are sitting there waiting for a signal from the antigen presenting cells in the cytokines to replicate and come back to clear the virus. We're also going to ignore some of the other T cell types. We're going to ignore helper T cells, which simulate B cells and antibodies. If we wanted to model antibodies, then we might not be able to get away with that. We're going to ignore memory T cells, which are responsible for the long-term immunity, because we're dealing with an acute infection, not secondary infection when you get re-exposed later on. We're also not going to talk about regulatory T cells, which are cells that come in and shut down the T cell response. Again, if we were talking about cancer, Tregs are quite important. One of the things cancers are pretty good at is persuading Tregs to come in and turn off the T cells that are killing them to allow the cancer to escape immune regulation. And so immune escape of cancer is one of the hallmarks of cancer and cancer progression. Once you have immune escape, you're in pretty bad shape with cancer. And so there's a whole set of really interesting questions. Again, and I keep coming back to this, several people are interested in doing projects on cancer. Specifically, some people have been interested in immune response to cancer. And we're not going to go into huge detail about the relationship between response to viruses and response to cancers, but I'll try to allude to it from time to time. And one of the things that's quite interesting is that, at least conceptually, the way your body responds to cancer cell and the way it responds to a virus could be totally unrelated to each other. There's no a priori logical reason why you have to have the same system responding to both things. They seem to be very different. And yet, the immune system has evolved in a way that allows it to use the same signals and the same cell types to respond to very, very different things. And if we were talking about response to bacteria, there would be yet another set of very different conditions which the immune system had to respond to. And so one of the things that's fascinating and frustrating about the immune system is its adaptability and its plasticity. And it also has this simultaneous ability to be extremely effective and then to fail in very complicated ways. Sorry, I need a little bit of water. And so we can dig into any of these issues more. From the point of view of modeling, there's so much we don't know. It can be sometimes a bit frustrating. If you're interested in bacterial infection, Denise Kirschner has done beautiful models of the immune response to bacteria, in particular tuberculosis, that are well worth looking at. We won't do that this semester. Maybe next semester we'll do that. Okay. So we're going to talk about the cellular immune response. Those were Sam's slides. This was my attempt to address that. I have a slightly different picture, a simpler picture. But again, the macrophages chew up either virus directly or fragments of cells that have died from apoptosis after being exposed to the virus. They turn into dendritic cells. They crawl to the lymph nodes. They form synapses with the T cells in the lymph node, stimulating production of those T cells. Those T cells come back And you'll notice that we don't just have one type of T cell, you have both the CD4 plus T cells and the CD8 plus T cells. And you have to have these interactions between the two to actually do the job. For the moment, we're going to assume that there's one type of T cell. And we're also going to do something else, which is not so wonderful, which simplifies our model a lot, which is we're going to assume that the signal that comes from the infected tissue to the lymph node is a single signal. And we're going to happen to call it cytokine, although it really should probably be called antigen presenting cell. And we really should have had two signals, one of which, because the time scales for those signals are different. The cytokine signal gets to the lymph node very quickly. The antigen presenting cells, because they literally have to crawl, can take days. And so combining those two signals is going to be one of the sort of the bigger compromises we're going to make in the name of simplification of this model. Although we've made so many simplifications, it's not clear that's the worst one we're doing, but it's something to be aware of. And then we're going to assume that those T cells come back from the lymph node, come into the tissue and kill the target cell. Exactly which pieces of this immune response we decide to model and how much detail we decide to model them in is a bit arbitrary to begin with. The paper of Erica Mohan and Bart Ehrmantraut does a very detailed model of this. The papers of Amber Smith do a very, very simplified, much more simplified model. So we're sort of in the middle. And the reason we're working, again, as I said at the beginning of this, with this not very elegant model that I wrote down quickly a few months ago, was because I wasn't finding models that were intermediate in complexity. I wanted to have a model that included cytokines, included the lymph node, included T cells, but was also not 50 equation, 99 parameter model. And so that's why we are where we are. So I want to talk, and this may take us till the end of the class. Having said all this, I'm going to talk a little bit about some principles of modeling. And this comes back to some of the ideas that I've talked about before, and it may seem a little bit abstract, but it's important to think about, and I think this is as good a place as any to introduce these ideas. We could come back to them later, and they may make more sense after we've done the whole process. But I do want to think about this. So in the simple model that we've written down so far, we wrote a lot of rate laws. We said that the rate of infection of a susceptible cell was proportionally beta times the amount of virus times the number of cells infected. That there was a transition rate K for going from I1 to I2, a transition rate D for going from I2 to dead, that the cells produced a constant amount per cell per unit time, P of virus. The virus was cleared at a constant rate. These are all linear relations. That is, they're or bilinear in the case of the first one. Otherwise known as mass action in chemistry. Now, there's no obvious reason why those relations should be linear. And when we start talking about the immune system, we'll find that if we use that kind of linear model, it doesn't work. It blows up in very unpleasant ways. Although if you look at the papers of Alan Perlson, and I'm always referring to Alan's work because it's so beautiful and elegant and so tightly grounded in experimental data, his models are almost always linear. If you look at his equations, they're not that many, and almost everything is linear. Occasionally there'll be one of what we call saturation term, but he tends to try to stick to linear equations as much as possible. And as we saw, we could actually reproduce the first couple of phases of our viral load curve with linear equations. We didn't need anything. And so you say, why are we going to have to introduce all this complexity now just to get that tail of the curve? And it's possible that we could come up with a set of equations that would get us the tail of that curve without going away from linear equations. But biologically, we know there are a lot of nonlinear things going on. And so if we were wearing pure mathematician hats, we'd say, what's the simplest possible model we could write that gives us that cutoff? We might be able to do that with linear equations. But since we know something about the biology of how the adaptive immune system works, we're cursed a little bit with that knowledge because we want to respect that knowledge in our model. And once we start writing rate equations, we already don't really know what beta is in detail. We've talked about that. The lifetime of the virus, we've estimated. Most of these things we can estimate. In other words, we know that the cells, once they're infected, they don't die in 10 minutes, and they don't die in a year. They last a couple of days at most. And so we can at least get a rough order of magnitude for most of these rates. When we're talking about things like how much cytokine is produced by the cells, and what's the rate at which the replication of the T cells is determined by cytokine, we don't have good measurements of that, or how the T cells come in and kill target cells. We're going to find that we don't have good experimental data for those. And so we have to think a little bit about what we do in the absence of detailed rate laws. And when you don't know what's going on in detail, the most important thing to do is to think about limiting cases. And the concept of a limiting case is what happens when some parameter or some variable is very small and very large. Because very often we can have a reasonable idea of what should happen in those conditions. So for example, if we have a susceptible cell going to an infected cell, and we have no virus, the rate better be 0. If we have a susceptible cell going to an infected cell and the amount of virus is very, very large, there still has to be some maximum rate. It shouldn't go to infinity. This equation does go to infinity. It tells us already that this rate law can't be right when the amount of virus is very big. But if we just focus on the 0, the simplest thing we can ask is, does the rate go to 0 as the variable or parameter goes to 0 or not? The rate of infection of our susceptible cells, if there's no virus, has to be 0. You can't be infected if there's no virus. On the other hand, there could be other situations in which we had a rule where the rate didn't go to 0 when a parameter went to 0. If there were no virus in the system at all, we could still have, for example, suppose our T cells died naturally because there was turnover of the T cell population. Then the rate of T cell death would be independent of the amount of virus. And so as the amount of virus went to 0, the rate of T cell death wouldn't go to 0. And so the first thing to think about is if your variable or parameter is 0, is the value 0 or not? And very often a simple logical argument will tell you most of the time the answer is it better be 0, but not always. Sometimes if something's inhibitory, your maximum is when your value, when your variable is 0. The second thing to ask about is the slope. Is the rate of increase of whatever it is finite at 0 or 0 at 0? If it's linear, it better be finite, straight line. If it's nonlinear, it could be 0. If the value can't go below 0, like the population of T cells of infectable cells can't go negative, then if I start at 0, I can't go negative. If I start above 0, I could go negative, 0 slope, or positive. Those are often things you can argue by looking at experimental data or looking at or just making a logical argument, just thinking about the problem. So you should always start out by thinking about what happens in any case when the value of a variable is 0 or very small. Again, and in all of these cases we've already gone over, if we have no I1 cells, then we have no conversion of I1s to I2s. And so the rate I1 goes to I2 has to be 0. Simplest way to get that is to write a rate constant K times I1. That's clearly 0 when I1 is 0. And so all of these linear forms come about from this kind of argument that if I have no I1s, the rate at which I produce I2s is 0. If I have no I2s, the rate at which I produce Ds is 0. If I have no virus, the rate at which I produce I1s is 0. If I have no Ts, the rate at which I produce I1s is 0. If I have no I2s, the rate at which I produce virus is 0. And so that very simple argument tells me that everything has to go to 0 at 0. And if my rate is not 0 near 0, assuming a linear form is the simplest possible thing I can do. In that limit of small values, assuming that things behave linearly is reasonable. For larger values, you could have more complex behaviors. And I haven't explained the Hill function yet, I don't think. We'll come back to those in a minute. But typically what we're going to find is that either we have something that looks like this at 0. Sorry, that's supposed to be a straight line. It's not a very good straight line. Or we have something that looks like this at 0. This one where we have an increasing curve that's nearly flat at 0 is going to be called a Hill function. And where it's linear at 0, we'll get what's called either a linear response or Michaelis-Menten kinetics. And that already tells us a lot. So if we know the value at 0 and the slope at 0, we can do a lot of estimation. So our first rule is that if we have a conversion of species like this, if the amount is 0, the rate's got to be 0. The second, since the rate can't go negative near 0 because that would mean we have a negative amount of the stuff, is that for small amounts, increasing the amount increases the rate. And that already tells us something about the rate law. So if I have a rate law for species X, which is f of X, is something arbitrary, any function, no matter how complicated. If I Taylor expand that function f of X around X equals 0, f of X is f of 0 plus X times f prime of f0 plus 1/2 X squared f double prime plus dot, 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 dot. Rule number 1 says that f of 0 has got to be 0. Because otherwise, at 0, all of these later terms are 0. So f of 0 is f0. So that means that first term disappears. Rule 2 says that f prime of 0 has to be positive, or my rate would go negative for small values, and I'm not allowed to do that because that would force my value to negative. And so that means that the highest order term I have is x times f prime of 0, which is positive. And if I assume x is small, all of these other terms are smaller. And if I neglect them, I get all these linear functions. And so that's the mathematical argument why you use linear functions. And in this equations that we've written here, we have 1, 2, 3, 4, 5. It says 4 here, but there are actually 5 of them. And so that's the basic mathematical justification for what we do. Now, if x isn't small, all of these arguments fail. Okay. Now, that applies fine for these simple transformations. I1 goes to I2, I2 goes to D, V dies, I2 is producing virus, all of those. In the case T goes to I1, it's a function of 2 variables. And if we'd written T plus V goes to I1, it would be clearer. Suppose that we have a rate law that depends on 2 species, in this case, V and T. What's the functional form? The functional form is done exactly the way we did it before. We Taylor expand around 0, 0 in this case. And then we get a bunch of partial derivatives. So we get f of 0, 0, plus x times df dx, evaluated at 0, plus y times dy df dy, evaluated at 0, plus 1/2 of all the second derivatives plus the order third. Our rule 1 says that f of 0 is 0. That means that f of 0, 0 has to be 0. What's more, it tells us something else. And this is actually typed wrong here. I'm sorry about that. I've got to fix that. F of 0, 0 has to be 0. What this should say is that df dx has to be positive, df dy has to be positive. I'm sorry. So I'm still wrong. I'm getting tired. I apologize. This is correct. Rule 1 says that if either species is 0, the rate has to be 0. That means f of 0, 0 has to be 0. But it also means that df dx of 0 has to be 0 and df dy of 0 has to be 0. Because this is dependent only on x, which would mean in order to be 0, the second part has to be 0 because x can be anything. The same thing here. This second one has to be 0 because these have to be 0 if x or y are 0. And if I set y to 0, that doesn't tell me x is 0. If I set x to 0, it doesn't tell me y to 0. So all of these initial terms have to be 0. There we go. Now, that leaves me with this second order term. And this one now, I have to look at this. The same thing is going to happen. This first term here, x squared d squared f dx squared, is independent of y. And that means it isn't 0 unless the derivative is 0. This third term here, y squared d squared f dy squared, is independent of x. That means that's not 0 when x is equal to 0 unless the second term is 0. So this first term and this third term have to be 0. And that leaves me with the lowest order term I'm allowed, f of x comma y, is 1/2 2 xy d squared f dx dy. That has to be positive. And that means that the lowest order term has to be what's called bilinear. That is, it's x times y times a constant. And that means to satisfy the rule that the value of either species being 0 makes the rate 0. And increasing either one gives me a positive slope, means that the lowest order result is xy times a constant. And that's what we have here, beta VT. And that's called mass action kinetics in chemistry. If people have had chemistry courses, you'll have seen some kind of derivation of mass action kinetics. You can derive it in a variety of ways. If you do it in a mathematical biology course, this is how they'll do it. Okay. So now we have that lower bound. And now we have to think about what happens for large values. Now, in reality, in biology, we never really have large values. Time doesn't go to infinity because we don't live forever. The number of the density of a particular molecular species doesn't go to infinity. Typically, molecular species concentrations are pretty small, unless it's water, maybe, or oxygen. The number of cells of a given type doesn't go to infinity because you could only fit a certain number of cells per unit volume. Nevertheless, we have to think about what our method does in the limit as things got large. Does it behave sensibly or not? And fundamentally, there are only three things that can happen. If our variable or parameter goes to infinity, the value could go back to zero. At zero, it could go back to zero. So it goes up, it goes down again. It could come to some finite value, or it could go off to infinity. In our linear case, the rate of cell death is k times the number of cells. As the number of cells goes to infinity, the rate goes to infinity. We have to think about slope. If the value is zero at infinity, it can't have a slope because once you hit zero, it stays at zero. So this has to be flat. If it's going to stay finite, it also has to be flat. If our rate goes to infinity as we go to infinity, the slope could keep going up. It could have some kind of exponential increase. It could be linear, stay straight, or it could saturate and flatten out like the other two. So we have a couple of possibilities. If we're converting cell types, if we have if we have cell of type A going to cell type B, the rates are independent. The rate at which one cell converts is independent of how many cells I have. And therefore, that linear function for conversion from I1 to I2 or I2 to dead makes sense. There's no reason that having more of them would slow things down. On the other hand, for viral infection, the cell can't get infected by virus more than once. And so if you have a lot of virus, you would expect that you don't have the rate of infection go to infinity. And so you'd expect to have some kind of saturation. In particular, if the virus is in the environment, it has to bind to a receptor on the surface of the cell and then be taken in. There's a rate associated with that. It takes time for that to happen. And that means that no matter how much virus I have in the environment, I can't infect myself faster than it takes for a virus to bind to the receptor and get pulled in. And so that's what's called a rate limiting step. Internalization of the virus is a rate limiting step. And in that case, we would expect that as we increase the amount of virus in the environment, the rate of infection slows down. And we get something like this picture here, where for small amounts of virus, the rate of infection goes up linearly, which is what we've assumed. For large amounts of virus, there's some maximal rate of infection. That's called saturation. When we have a saturation curve like this, it could have a lot of different shapes, but typically the things we care about are, we know the value at zero. The only thing it has is a slope at zero. We have the maximum value at infinity, and then we need to know how steeply it curves over, and that's typically defined by the value at which it's half maximal. And that number is usually called K, capital K. There are a lot of reasons you can have saturation, but processes that take a finite amount of time are a common cause of saturation. If a cell takes some amount of time to die, if the virus takes some amount of time to reproduce, if the cell takes some amount of time to reproduce, if it takes time for the antigen presenting cell to crawl to the lymph node, anytime that something takes a finite amount of time to happen, we expect not to see linear responses, but to see saturated responses. And that's something that's a very important concept that we'll go over. And when you read papers on modeling, that's something that's implicit in almost everything that's written down. And it's not presented, it's sort of assumed that people have been through this kind of argument. Maybe it's better to learn by doing it rather than have it presented in an abstract way. But you'll see in papers all the time, we'll use the Michaelis term, which is a saturation term. We'll use a Hill function, and they won't really explain why. And so I want you to have some background about the thinking that goes into those decisions. Okay. If I want to have a curve that looks like this, the simplest form that I can write is this one. F of x, if x is the x-axis here, is equal to v, which is called the maximum rate, times k. I told you capital K would show up, which is that half maximum value, otherwise known as saturation constant, x divided by k plus x. It's nice to rewrite that by dividing through by k, in which case f of x is equal to v times x divided by 1 plus x over k. What does this do? If x is small, in particular, if x is much less than k, then this denominator is effectively 1, and f of x is equal to v times x. That means the slope at 0 is v. Okay? That's a critical argument that we'll need again and again. If x is very big compared to k, much bigger than k, then this 1 doesn't matter, and I get v times x divided by k, and I'm left with, it should say v times k, I'm left with v times k. And so then the maximum value is independent of x, it's constant. Okay. There are other functional forms that I could write that would do the same thing. I could have an increasing exponential that would work as well. It looks pretty similar at 0 and infinity. Its shape near the halfway point is a little bit different. I might also want something which is the flip of this, where I start not 0, wind up at 0 at large values, and my slope is non-zero decreasing at the beginning. And that is easy to do. Instead of putting an x in the numerator, I put a 1 in the numerator. And then in that case, I clearly get if x is small, I get v here. If x is big, I get v over x times k, so I get v times k over x, which goes to 0, so it goes like 1 over x. So it goes like 1 over x. If I wanted to have that, the flip of this increasing exponential is just a decreasing exponential. If I want to mix multiple species together, I can do that in a whole variety of ways. I could multiply terms like this. I could add terms like this. I can combine them in a variety of ways. And one of the things we're going to run into pretty soon is that we're going to have to combine some rate limitations, and we're not really going to know how to do it. And the biggest problem we're going to have with the model we're going to build here is going to be that the way we combine the rate limitations isn't quite right. Okay. So in particular, we're going to run into this in the context of killing, because we're going to have CD8 plus T cells that are going to be in our tissue, and those CD8 plus T cells are going to kill our infected cells. And the simplest thing to do would be to say that the killing rate is proportional to the number of infected cells, which makes sense, and the number of killer cells, which also makes sense. However, that means that if you have a few immune cells and a lot of infected cells, the rate of killing should increase linearly as I increase the number of infected cells. But if I think about how killing occurs, that can't be right, because a T cell actually takes time to kill a target cell. So when I have an effector T cell that binds to an infected cell, it takes typically on the order of 40 minutes to kill its target. That could be off by a factor of 2. And that means that each T cell can kill about 30 cells per day target. And that is a problem if I assume that there are a few T cells and a lot of infected cells, because a linear relation would say that I can, if I have 10 to the 7th infected cells and I have 10 T cells, it's going to tell me that I'm killing lots and lots and lots of infected cells per day. But in fact, 10 T cells can't kill more than 30 infected cells each. 30 times 10, 300 infected cells a day, independent of how many infected cells I have. And so I'm going to expect that I get this kind of saturation. And this kind of saturation has a name in chemistry. It's called the Michaelis constant. It's a Michaelis-Menten relation. And the K, which is the half maximum saturation number, is called the Michaelis constant. And so you'll sometimes, and the reason I'm saying this is sometimes you'll see different names for that K factor. You should always write rate laws so that saturation terms go from 0 to 1 as a function of X. So this K divided by K, if X is 0, it's 0. If X is very large, I get K back. So I actually should have divided by K. I shouldn't have the K in the numerator. I wrote it wrong. So I've got to fix that. Okay. So in the example I gave here, if the immune cells can kill 30, if one T cell can kill 30 infected cells per day, then as a function of the number of immune cells, the number of infected cells that I kill should be at most 30 times the number of immune cells, except the number of infected cells divided by 1 plus the number of infected cells divided by the number of immune cells. I'm not saying that's the right relation, but that's the kind of argument that one can use to make this kind of case. In the absence of knowing what's really going on, these are kinds of arguments people use. Maybe I should stop there, Josh. Do you think that's enough? I think trying to do Hill functions in 5 minutes is probably going to not work. So that gives us some arguments. There was a typo because of the Ks. Josh, you can help me go through and make sure that I fix those Ks, get rid of the Ks where they don't belong and put them in where they belong. And I appreciate your patience going through some things that can look pretty abstract. I don't really know a way to introduce some of these concepts without doing a little bit of abstract reasoning first. When we come to actually doing the modeling that we're going to do, we're going to find that we need to use saturation terms a lot. If you read the typical modeling paper, those saturation terms are pulled out of the air. They'll just say, we use the Michaelis form. And they don't give any justification for it. And there actually are good mathematical and biological reasons why those are the first forms you reach for when you have ignorance. And again, the main reason is that what happens at zero and what happens at infinity. What happens if you have a small amount of the thing and a large amount of the thing. And so that's, I guess I'll open it up. We have 5 minutes for questions. If that didn't make any sense, let me know and I'll try to explain it more clearly next time. It may be clearer when we apply it. I also would refer you to any of the standard mathematical biology textbooks which derive Michaelis-Menten, but it's usually derived in the context of biochemical reactions. And it's said that in chemical reactions, they have this form. And then, by the way, we use it in all of these other contexts. And the real justification for doing that little derivation I did with the Taylor expansions is that it's not specific to chemistry. It's a property of the mathematical structure of any continuous differentiable function, which is 0 at 0. And if I made the same argument for boundedness, I could show the same argument. Okay. Any questions or comments before we break? Everybody's probably pretty tired today. I talked a lot and we only did that one exercise. So again, I will try to redress the balance and not speak so much versus doing things. But having gotten through this, I think then the derivation of the functional forms that we're going to use will make a little more sense. Okay. Anybody have anything you want to talk about before we break up? Last minute. People are probably tired and hungry, huh? Josh, you're probably hungry. You already had dinner, right? You ate during the lecture. I hope some people had dinner during the lecture. That would be all right. Okay. Okay, well then, thank you. I appreciate your showing up. And I really do encourage you. I really would like to see some small simulation result next week. And since we didn't get through the exercises that I had planned, there won't be exercises for the homework, or at least at most one small one. And so that means that you can focus on working on your projects. But I would like everyone to do a small presentation of what they've gotten done next week. Again, it doesn't have to be slides in detail. If you want one slide, that's fine. But I would like to see everybody have some notebook which has some running simulation of what you're going to work on next week. And if you want to talk to me about that, that's fine.